Good evening, everyone. I'm just going to wait a minute for all the audience members to file in to the webinar. Welcome, everyone. We're so happy to see you joining us. I'm going to start now. My name is Dr. Janive Remy. I am the director at the Sridas Ramphal Center for International Trade, Law, Policy, and Services. And it is my esteemed pleasure to welcome you all to this webinar tonight on the science of the COVID-19 vaccine, the role of the university, which I have the pleasure of co-hosting with the University of Guyana, as well as the Cave Hill Faculty of Science and Technology. When I was first approached by the Chancellor of the University of Guyana to assist in putting on this wonderful webinar, it didn't initially occur to me that this was something that would fall within the exact remit of my center, which deals really with trade and trade policy. But as I learned more about the contents of the webinar, I thought it would be an excellent opportunity for Cave Hill, the Cave Hill campus, to be involved in a webinar of this nature, which is really about, one, explaining the science of a vaccine that has been responsible for saving millions of lives around the world. But more importantly, in my view, having the opportunity to celebrate a son of the Caribbean. This webinar is really it's really being held around and to celebrate the work of Mr. Vidya Rupchand, who is the principal research scientist at Pfizer, the company responsible for the vaccine, COVID-19 vaccine. And we are so pleased that we are able to collaborate on this venture and introduce him to a Caribbean audience, as well as bring in some voices from across the Caribbean and from the university to speak about the importance of these types of innovations and what role the university can play in the Caribbean in ensuring that these types of works are able to be propagated from the region. It's my pleasure, before we turn to the actual evening, the man of the evening, to ask Professor Eddie Green of the University of Guyana, the Chancellor, to also warmly welcome you to tonight's webinar. Professor Green? Professor Green. Professor Green, you may turn on your video and your mic. Yes, thanks.
Can you see me now? Hello?
Yes.
Yeah.
Yes.
Okay. It is indeed my pleasure to participate in this webinar, which hinges on three pillars. First, it is an inspiring story of a Caribbean student graduating from a Caribbean university, the University of Guyana in particular, grasping opportunities in the metropolis and excelling to the point of being a leading global light in the global vaccine development. Second, it is about the importance of science, the evolutionary processes and the extraordinary innovation pathways that contribute to sustainable human development. And third, it is about the aspirations of our Caribbean universities working to break new barriers and establish the enabling environment for grooming current and future scientists to develop their potential to the fullest, and at the same time contribute and consolidate and advance the region's capacity to compete in the international arena. I am sure that the discussion this evening will help us to better understand how human, technical, and economic resources can be marshaled toward a fusion of science with the delivery of health services, therefore improving human conditions. That the Sridas Ramphal Research Center, whose CEO, Dr. Janive Remy, so willingly agreed to facilitate this event, that on our panel, we are honored with the presence of Professor Clive Landis, principal of the UWI Cave Hill campus, himself a renowned scientist, and Dr. Tomlin Paul, the former dean of the Faculty of Medical Sciences and a pioneer in the what you will hear later on, the H3EC, what I call the Caribbean Institute of Genomics. And this gives you an idea of why and how UWI could be ranked by the Times Higher Educational Supplement among the top 1.5 universities in the world. We celebrate that. But together with the participants and the panelists is our esteemed moderator, Dr. Janice Bedok, Dean of the Faculty of Natural Sciences, UWI Cave Hill, and our feature speaker, Mr. Vidya Rupchan, Guyanese Pfizer expert. And of course, the participation of this virtual audience from around the globe. I'm sure this will no doubt help to make this webinar a memorable event. I thank you for your attention.
Ladies and gentlemen, tonight I am pleased to welcome 3 panelists with extensive research and administrative experience, which will be brought to bear in a comprehensive way in this forum. The highlight presentation to be shared by Mr. Vidya Rupchan will take us on the journey of the discovery, launch, and approval of the Pfizer-BioNTech COVID-19 vaccine, an mRNA vaccine which represents a new age in vaccinology designed to fight the SARS-CoV-2 virus. But his personal story is no less enthralling. It began right here in the Caribbean, born, raised, and educated in Guyana, before migrating to the USA and ultimately landing at one of the largest pharmaceutical companies in the world and right in the middle of the COVID fight. For me, it is that journey that will keep me glued when he begins his presentation, which I know will also focus on how his early Caribbean training prepared him for this inevitable path. The audience will also hear from Dr. Tomlin Paul, who will connect us with the importance of Caribbean science and its contribution to societies, and present the work being done at the under the H3E initiative to build Caribbean capacity and representation in global genomics and health. And finally, on the topic of the role of the university and its contribution to the advancement of science, our own Pro-Vice Chancellor and Principal of the Cave Hill campus, Professor Clive Landis, will provide remarks about the leading historical contribution of the UWI to science and the emerging areas where we are poised to make an impact. But before we begin, permit me to tell you a little bit more about each of our speakers. Mr. Vidya Rupchan was born and raised in Guyana and received his undergrad degree in chemistry, you'll notice my smile, from the University of Guyana in 1990. He migrated to the US, where he received a master's degree in immunology from New York Medical College and a second master's degree in chemical engineering from Columbia University. He is currently a principal research scientist at Pfizer, where he has been for the last 28 years. He has spent his entire career in the vaccines division, and his efforts have contributed to the reduction of viral and bacterial diseases around the world. He was part of the Kick While Polio Out of Africa movement, which was initiated by the late great South African president, Mr. Nelson Mandela. Mr. Rupchan was also part of the Pfizer-BioNTech team, which delivered the first COVID-19 vaccine, BNT162b2. Our second speaker, Dr. Tomlin Paul, is a family physician and a medical educator. As you heard, he's a graduate of the University of the West Indies with training in public health and epidemiology from the Johns Hopkins University School of Public Health. He has taught and conducted research in community health and medical education at the University of the West Indies, and most recently was Dean of the Faculty of Medical Sciences at the Mona campus. He is an avid advocate for social accountability in health professions education, and is currently the co-lead of the H3E Caribbean Human Hereditary Environment and Health in the Caribbean Initiative. Our final speaker, Professor Clive Landis, who joined the UWI in 2004 after relocating from Imperial College London, has served as Deputy Principal of the Cave Hill campus, as University Pro Vice Chancellor for Undergraduate Studies, and Chair of the UWI Zika Task Force in 2016. He is also currently Chair of the UWI COVID-19 Task Force, having assumed that role since February 2020, and recently has assumed the principalship of the Cave Hill campus for a period of 5 years with effect from August 1st of this year. An immunologist by training and in practice, Professor Landis has published and presented extensively his research, which has focused on the role of inflammation during wound healing, vascular disease, and recovery post-surgery, and recently has evolved to include public health research on viruses such as HIV, ZV or Zika virus, and of course, SARS-CoV-2. So let us begin with our first presentation by Mr. Vidya Rupchan, who will tell us about the science of COVID-19 and the vaccine. And then we're going to pause briefly from some questions from the audience before inviting Vidya to tell us more about his personal journey from the University of Guyana to Pfizer. Vidya, the floor is yours.
Well, thank you very much, Dr. Bainrock and everybody from UWI. It's a great pleasure to be here and an honor to be representing Pfizer and, of course, my Guyanese people. The talk is about the science of the vaccine and the history of how they were different disciplines and how they came together to make this vaccine. So even though you hear the term vaccine, what probably comes to mind is immunology. Rest assured that immunology was the last part of this. It started with organic chemistry. And as an undergrad in chemistry from the University of Guyana, I always have to emphasize that what we do here starts out with organic chemistry. So I'd like to give a shout out to my chemistry education, first of all. Okay, so let's get started. So on your left here, this is what the virus looks like. If you look here, there's a spike, and that spike looks like this. It has a receptor binding domain on the top, and then the rest of it is in the cell membrane. So keep an eye on the shape of this spike because it looks like a lollipop, right? So this spike here on the outside of the virus looks like a lollipop. So this is where it binds to the cell. If you're not a virologist, don't worry, we'll go through it a little bit. So what happens, how does this virus enter a cell? Well, the virus binds to ACE2, which is angiotensin 2 converting enzyme. And what happens once it binds to ACE2, there's another protein on the surface of a cell called TMPRSS2, which clips it. Sorry. Once it's clipped, it goes into post-fusion conformations. So it goes from this lollipop, which is what the native virus looks like, to this more linear as it attaches to the cell. So that's very important because the vaccine that we made is something that's based on the pre-fusion conformation and not the post-fusion conformation. So we want to make something that looks like this. So later on, I'm going to show you all the tricks that we use to ensure that we locked it into this pre-fusion conformation. So that's a little bit of virology for you. So the Pfizer-BioNTech vaccine is an mRNA vaccine. And the mRNA vaccine uses synthetic mRNA. So this entire vaccine is synthetic. So as a synthetic chemist, I loved being part of this because everything was about chemistry at the start. So the Pfizer-BioNTech vaccine and the Moderna vaccine, which is the other RNA vaccine, use the synthetic RNA that contains information about the coronavirus signature spike. So the spike protein is 1,273 amino acids long. So the mRNA codes for 1,273 amino acids. And how do they work? So they work by introducing an instruction that directs the body to produce small amounts of the spike protein. And once the immune system recognizes this protein, it starts making antibodies and T cells, which is what a vaccine does. A vaccine causes your immune cells to make antibodies and T cells. So this is basically a summary. We made the mRNA, we injected the mRNA, and we produced T cells and B cells. Okay, so the mRNA that we made, and that's how mRNA works, it has to be capped. So you have to trust me on this because I can't go through the whole capping mechanism. The virologists out there would know what the capping is. So at the 5' end of the mRNA, there's a cap, and that cap is there to stabilize the mRNA, and it allows the translation of the mRNA into protein. And there's an untranslated region at the 3' end of the mRNA, and that helps to stabilize it. And there's also a polyadenylated tail that mRNA has to have, or else it doesn't get translated. The cap1 that we use on this mRNA reduces the activation of pattern recognition receptors. And those are basically a part of your innate immunity, which your cell has. So your cell has evolved, cells have evolved over the years to recognize viral proteins. So the cap1 we use is similar to a cap that's used that a mammalian cell would use. So when your innate immune system sees the cap1, it thinks it's a mammalian mRNA. So it's a way of sneaking past your innate sensors, like your MDA5 or your RIG-I or your TLR3, your TLR7, and your TLR8, which are toll-like receptors. I see Professor Land is shaking his head. He probably knows a lot about innate immunity. So the one thing that we started with was this cap1, and the cap1 is similar to a mammalian cell cap. So remember that, right? So there are a lot of tricks that we had to use to ensure that our innate immune system did not shut down the protein synthesis. Okay, so how do we do the other trick? So one of the bases that mRNA uses is uridine, and mammalian cells usually stamp a recognition pattern on some tRNAs. And a lot of research was done by Kathleen Carrico, you probably heard the name a lot, and Drew Wiseman at the University of Pennsylvania, and they showed that pseudouridine, which is modified uridine, is a way of evading your innate immune system from recognizing mRNA. So what we did is that we took it a step further, and it's also been shown that pseudouridine can be further modified into methyl pseudouridine. And so when methyl pseudouridine is incorporated into mRNA and mRNA gets into your cell, your innate immune system thinks that that's its own. So it doesn't try to shut it down. So it sneaks past the TLRs in the endosome, the TLR3s, the TLR7 and TLR8. And if some of it ends up in the cytoplasm, there's the RIG1 or RIG-I, as some people call it. There's MDA5, there's LGB2 that think that this RNA is self. So these are the two tricks that were necessary to ensure that the innate immune system did not recognize the mRNA as foreign. I don't know how many of you have seen that movie, The Matrix, but you remember that submarine, the Nebuchadnezzar, that was using all this shielding tricks? That's a great movie to see, but that's what we did. We basically cloaked the mRNA by using a pseudouridine and a cap. So that's how the mRNA stays into the cells and does not get degraded by innate immunity. Okay, so I want to give tribute to all these great West Indian organic chemists who have influenced me. So this is basically the synthesis of a pseudouridine and methyl pseudouridine. So on this side, there's Ulrich Trotz, who created this chemistry program at the University of Guyana. This is Wilfred Chan. I think most of you know him from UWI. This is Dr. Winston Tinto, who was my chemistry professor at the University of Guyana, and then he became a professor at Cave Hill. This is Vernon Box, who was also a professor at UWI, and he taught me organic chemistry when I first came to the United States. So I have a history with all of these guys. I mean, I didn't directly interact with Wilfred Chan, but he interacted with Dr. Trotz, he interacted with Dr. Tinto, and he interacted with Dr. Box. But these were the giants of organic chemistry that I kind of grew up with and who influenced me. I mean, Dr. Tinto was my final year organic chemistry teacher, and a lot of this synthesis he would love. But this is basically how you make pseudouridine. I wouldn't go through all the steps, but once you make pseudouridine, you can use it as a pseudouridine, or you can methylate it. So this is the methylation here, where you go from pseudouridine to methyl pseudouridine. And I'm sure, you know, Dr. Trotz and Dr. Box and Dr. Tinto could all synthesize, and Dr. Badenak as well could synthesize methyl pseudouridine. I see she's smiling there. But this is the hardcore organic chemistry that was necessary to make this vaccine. So don't think that biologists alone make this vaccine. Don't ever think that. There were chemists who were able to synthesize large amounts of these bases. All right, so we went through the modified bases. So I kind of touched on this before, but the presence of this modified nucleoside in the mRNA suppresses the immune activation. So your TLRs think itself. And then because it's stable, you know, it stays around a longer time and you get more protein, which means you get a stronger immune response. And by deimmunizing mRNA, you broaden the therapeutic application. So this technology can be used for other things later on. And I talked about these TLRs, the toll-like receptors, they think that itself, so it's dampened. So that's how you sneak the RNA in. But, you know, I just wanted to ensure all the organic chemists in the world realize what an important role they had to play in making this vaccine. I don't want these biologists to think that they alone made the vaccine, because that's not true. All right, so this is just a little bit about the receptor binding domain. So we talked about the receptor binding domain. So that was just a part of the virus. But we use the full-length spike. So we use the full-length spike protein to make the vaccine, because you get more antibodies and T-cell epitopes because you have a larger protein. So therefore, the COVID vaccine that we made, the BNT162b2, is the full length, not the receptor binding. So this is just some dimensions. You know, I know chemists and biologists love that dimensions. So the total length of the spike protein is 1,273 amino acids. Okay, and the receptor binding domain is 222 amino acids. So the receptor binding domain is like a sixth of the total spike protein. Okay, so this is the money shot. So remember I said that the spike protein is 1,273 amino acids. So when the virus is in a native form, it's got this pre-fusion conformation that looks like a lollipop. But remember I said when it binds to the cell, it goes into the post-fusion form. So what we want to do is lock this virus into the pre-fusion form. So we want it to look like a lollipop. So a lot of work was done on other coronaviruses starting in 2003 with when the first SARS came, and then MERS later on. So people have been working on a method of locking this thing in this pre-fusion conformation. And what they found was if they did a lysine to proline mutation at position 986, so this is how you write it for you non-biochemists over there. So it's K986P, means is lysine is mutated to proline, and then a valine is mutated to proline at 987. They realized that if you did those two mutations, because proline is so rigid, remember this is proline, there's very little movement in proline. It's so rigid that it locks the viral protein in the pre-fusion conformation, so it looks like a lollipop. So if you transfected cells with this with a 2p mutation here, so this here right there is where the 2p mutation is. So if you transfect the cells with RNA that contain the pre-fusion conformation with the two prolines, and then you purify the protein, you can see that it still looks like a lollipop. So here we are sure that the 2p mutation locked the pre-fusion protein into the pre-fusion state, so it can't flip into that linear form that you saw in the first picture. So this is the money shot basically, and I would like to, you know, especially acknowledge all my chemistry professors at UG, Dr. Halvidar Singh, Mr. Malcolm Bumbry, Dr. Winston Tinto. He was the guy who introduced me to amino acid biochemistry back in 1990 for our 401 chemistry class. So these two mutations is what allowed us to lock the protein in the pre-fusion form. That's one of the key things that made this vaccine so successful. All right, so this is some history because I want to tell people and ensure that everyone understands that SARS and MERS allowed us to learn about coronaviruses. So we didn't just get up one morning, you know, 2 years ago and say, let's go do this. People have been working on these viruses for a long time, and HKU1 is also coronavirus. And so people have been looking at these mutations. You see what these mutations are? They've been looking at different mutations and they've been looking at how to stabilize the protein. So here, if you look at this, these are the wild type MERS, and this is the wild type SARS. And if you did the 2P mutations, it would be this and this. And if you look at the way they behave, this is basically, this is basically an HPLC. So the more globular proteins have the 2P mutation. So that's why it's locked. The linear ones do not have the 2P mutation. So they don't show this globular nature. And you can see if you did X-ray crystallography, this is the MERS wild type. So you can see some of it is in the prefusion conformation, like that lollipop, but some of it is linear. So that would not make a good vaccine because it's gonna, by the time you put it in the vial and you let it sit at 4 degrees, it'll flip. So if you lock the MERS protein with these 2P mutations, like I showed you earlier, it stays in this globular. So this is very important because I know a lot of people are concerned, how come we made this vaccine so fast? We made this vaccine so fast because we knew so much about it. And a lot of groups, you know, X-ray crystallographers, biochemists, molecular biologists, all contributed to the amount of knowledge that we have in the public domain, so we could all move very rapidly when this virus appeared. Excuse me. So that's how the mutation was learned. I think if we didn't have this background from SARS and MERS, we still would be knocking our heads against the wall because we wouldn't know what to do to lock it. But people have been working on it for almost 17 years. Okay, so let's get to the vaccine now. So the mRNA vaccine has to be protected because mRNA is very labile. As Dr. Landis said, you look at mRNA, it degrades. So how do we protect this mRNA? Well, you protect it with a lipid nanoparticle. So these lipid nanoparticles are made synthetically, and we have a process on which we lower the pH. And once the lipid particle lipids get ionized, they become positively charged, and then they bind to the RNA, which is negatively charged. So it happens very rapidly, and that's why it's called a nanoparticle. It happens in nanoseconds. So this is the thing, that the structure of the RNA, like we went through before. There's the cap, the 5 cap. It looks like a cap. There is the receptor binding domain with the 2 regions of the spike, and there are the 2P mutations, the K986P and the V987P. So this is the vaccine. It has these structural and functional lipids, and it has the RNA, and that's how, in case Professor Landis didn't know, this is how we protected the RNA. We use these lipids. So there was a lot of organic chemistry that has been going on for a while now for delivering pharmaceuticals using lipids, but it was mostly for beauty products, for people who wanted their skin to shine, or they wanted these fancy ointments that are going to last a lot longer. So a lot of this lipid stuff came from beauty product chemistry. So I guess beauticians would be proud that they had something to do with it as well. All right. So these are some of the lipids that we used. They're called ionizable lipids because this is a tertiary amine. I'm sure all these chemists there would recognize the tertiary amine, so it can get ionized. So once the tertiary amine gets ionized, it gets a positive charge, and then it binds to the lipids, to the RNA. And I bet Dr. Box, Dr. Tinto, and Dr. Trotz, they could all write out synthesis of these lipids. And I'm sure they all have their own unique starting point. But I just want to acknowledge, these are great guys. These are great chemists, great scientists that came out of the Caribbean. And what I am doing now is directly as a result of the education I received from these great giants of organic chemistry. I stood on the shoulders of these giants because they really inspired me, and and I think the Caribbean has to be proud of where we are in this effort, but not just of me and the face. But I didn't get it by myself, so I can't stop acknowledging all these professors who had an impact on me in Guyana and in the US, because Dr. Box taught me at City College when I first came here. All right, so this is how you manufacture the vaccine, and this is where the chemical engineering comes in, right? Because we went through the organic chemistry, right? We went through how you synthesize the methyl pseudouridine. We went through the lipids. We went through the molecular biology, the arrangement of the RNA. cap and the 5' region, and then the polydinylation. That's all molecular biology. So now we have to make billions of doses, and of course, you can't make billions of doses in a small lab. So you need this big factory, and the way you make the RNA is you make it in DNA. You have to make DNA. So you make a DNA template that encodes the RNA, and then there's transcription. So there's in vitro transcription. So you go from DNA to this transcript. You make RNA. Then you have to purify the RNA. So here is a very important step, because remember, RNA can induce your innate immune system, especially if you have small hairpin loops. And if a small hairpin loop appears in your cytoplasm, your innate immune system is going to shut it down, because there should be no hairpin loops in the eukaryotic RNA. So that's why it has to be very, very pure. If this RNA is not very, very pure, the vaccine won't work, because your innate system is going to shut it down. So that's why there's an HPLC in part involved, where we purify the RNA before we formulate it. And then the drug substance. So this thing is made in 3 different sites. So the RNA is made one place, then the formulation is done in another place, and it's filled in another place. So this is the drug substance. That's just RNA. Then we take the RNA, we mix it with the lipids, with the ionizable lipids that you saw earlier. Then we make the drug product, and then that's the vaccine. So this is the process. So how does the vaccine work? So we went from organic chemistry, immunology. So everybody is here in their discipline, right? Organic chemistry first, right, Janice? Organic chemistry, molecular biology, chemical engineering, and now immunology. So how does this RNA work? So it causes a local transient inflammation. So once the protein is made, the antigen-presenting cells, like your dendritic cells, your neutrophils, they all come to that site where the inflammation is occurring, and they all pick up the RNA, and then they take it back to the lymph nodes. And in the lymph nodes, you get priming. So your T cells and your B cells start stimulating each other, and you get all this immune response. So you get antibodies made by B cells, and you get T cells getting primed by these MHC molecules. All right. So I have Dr. Trotz again. This is one of my favorite slides. And Dr. Trotz is a huge believer of this philosophy. Organic chemistry gets translated into biochemistry, but gets translated into immunology. So it starts with organic chemistry. But this is the immunology part. So this is a schematic of how it works. So you take your RNA, COVID spike sequence, you have your synthetic RNA, you make the lipid nanoparticle, and then you inject it. And when you inject it, the RNA gets translated. This is the biochemistry part now. So I'm not neglecting biochemists and molecular and cell biologists. So once that gets translated, you get your spike protein. So some of that spike protein gets chopped up, and it goes into endoplasmic reticulum. It binds to the MHC class 1 molecule, and then it gets transported to the surface of the cell, where it primes your T cells. So this is where your T cells get primed. So that's a T cell response. Or some of it gets transported to the surface of the cell, where it's recognized by B cells. So B cells produce antibodies. So the B cells produce your antibodies. We can neutralize the virus, and your T cells can kill virus-infected cells. And this is uridine and pseudouridine. We went through that before. So the BioNTech Pfizer vaccine is 162b2, and the Moderna one is mRNA 1273. So they're both the same technology. Okay, so that's how the vaccine works. And this is the functional assay. So this is a neutralization assay. What this basically shows is how the virus produces antibodies that can neutralize a swath of a different SARS-CoV-2 strain. So this is the USA Washington strain, or the Wuhan strain, what the vaccine was based on. So you can see it neutralizes this virus very well. So this is a log 2 scale. So what these numbers really mean is that if you diluted the serum from an immunized person 1 to 532, it'll neutralize the virus, right? So this vaccine neutralizes the Washington strain, which it should. It also neutralizes the Brazilian strain, the UK strain, and all of these other variants that came out, the South African strain. And it does, I don't have the Delta variant data here, but it also neutralizes the Delta variant. So it's a very potent vaccine. And we went into the clinic in July, and then the data came out in November, sometime in the middle of November. And that's the story. And this is the timeline of how it was made. We started talking about it in March of 2020, and then we went into the first safety studies in May, and then all the way to here, November. We got the authorization from the US, and November the data came out, and we got UK emergency approval and US emergency approval, and now we have full approval for adults and certain kids. And that's the story of the vaccine. It's called Comirnaty. And this is my favorite picture after the organic chemistry one. This is the Pfizer vaccine arriving in Guyana. So all my Guyanese kids are getting the Pfizer vaccine, and everybody, it was a very fulfilling experience for me, because it just goes to show that even though everybody's face is not attached to this vaccine, they all had input in my life. So I like to acknowledge everybody who has had input in my life. And for more information, you can go to this website. They have everything there. I'd like to thank everybody, my parents, my grandparents, my wife, my kids, my mother, my teachers, everywhere, my primary school, University of Guyana, New York Medical College, Columbia University, my friends, my relatives, Pfizer, and of course, people at UWI for reaching out to me, and so that we could all share this story. And are there any questions in this part? Just directed to me. This is me here. And this is me in 1977. That's my great-grandfather's house in Guyana. So that's the story. I mean, I tried to give you a not so detailed presentation, because I know there are a lot of non-scientists and I don't want to bore them, but we can talk more later on. All right, thank you.
Thank you, Vidya. Thank you for that first part. It is just that, the first part of an exciting afternoon. Vidya, here's what I heard. I was so impressed. I was here smiling like a crazy woman when I was hearing about the role of the organic chemist. So I heard interdisciplinary scientific team. I heard organic chemists. I heard molecular biologists, biochemists, immunologists, crystallographers, lipid chemists, chemical engineering. Now I got a question for you before I take some of the ones in the chat. Where is, if there is indeed, where's the role of the machine learning scientist, the modeling? Did that have a role to play in drug discovery, in vaccine production?
That was probably used by the X-ray crystallographers when they were doing those mutations, the 2P mutations. They probably modeled several mutations, because I don't think they went through 1,273 amino acids and just mutated them one by one. They probably did a lot of in vitro stuff, in silico stuff before they did it. But proline is usually the amino acid that most people go to when they want to lock something, because proline is like crazy glue. But I'm sure there was a whole lot of machine learning earlier. But by the time it got to us, that's why I say, a lot of work was done since 2003. So by the time it got to us, a lot of that stuff was published and it was public knowledge, but we just used the information because we were under the gun.
Okay. So we
but
definitely,
there's a lot more. Sorry, go ahead.
No, I was just stressing the point that there really is a role for scientists, all types of scientists, in drug discovery and vaccine discovery. And so you're kind of proving that when you're showing us all the different aspects that they contributed to.
Yes, and the other thing I want to say is that people have been trying to call this field something, they're like, oh, it's synthetic biology, or it's bioorganic chemistry, or it's chemical biology. And I try to call it vaccinology. I said, it's vaccinology, because there's no way you can call it chemistry, or you can alone, because, the chemists can synthesize things, right? But it needs to get into a cell, and immunologists have to read it, and molecular biologists have to clone it, the gene, into a plasmid. So, and chemical engineers have to scale it up, and clinicians and statisticians have to run the clinical trials, and physicians have to give the shot. So, I think if you ask me, which is more important, they're all important. There's no field here that is less important than the other, because they all have to work together to make a vaccine.
Good. So we have a couple of questions for you, Vidya. I hope we have maybe a couple of minutes, and then we'll turn back to you to do the second part. So the first question, is the spike protein pathogenic, and can it get into the bloodstream?
No, the spike protein itself is not pathogenic, because the virus is pathogenic, but the spike protein is just part of the virus. So you're not getting any toxic effects of a spike protein by itself, because what we are introducing is the RNA that codes for the spike protein. And that spike protein goes to the cell surface, either as a peptide in an MHC or as a whole spike protein, where it binds to your B cells. So it gets expressed on cells, but I don't think there's any evidence of it, getting into your bloodstream and spreading.
All right, thank you, Vidya, for that. A couple other questions. What was the quantitative yield, if you do know it, associated with the assembly of the lipid nanoparticles that necessitated a second stage of purification? That one went very technical.
I don't know that. I had nothing to do with the formulation. And even though I did know, I couldn't tell you. That stuff is proprietary.
Okay. Let's try this one instead. How many plasmids did you go through? How long did that process take? Do you know?
No, I don't know. But remember, I said before, a lot of people were working on other coronaviruses. So they kind of optimize it by the time we got it. But there are certain sequences and plasmids that are proprietary that we use, but those can't be revealed in this forum.
And then how about this one? What is the role of the virologist? And it's an interesting question because we didn't actually call the title virologist. So what's the role of the virologist? And is there an advent present in the nanoparticles? Forgive my pronunciation.
What's the last part?
Adjuvant.
Is there an adjuvant? No. So I could show you what's in the nanoparticle just now. Of course, the virologists are present because it's a virus vaccine. So the virologists have to grow the virus, which is what I do. So I grow the virus. So I've kind of evolved over the years. So I started out with the polio vaccine. I was growing viruses, and then I was working in immunology. But I mean, that's how you know if your vaccine is working. The virologists have to grow the virus, and they have to run the assays with the immunologists. In terms of adjuvant, no, there's no adjuvant. The vaccine is basically, phosphate, some salt, some sugar, the mRNA, and the lipids. That's the vaccine. There's no adjuvant.
And then, how long did it take to make the mRNA?
It took it depends on what you're talking about, right? Is it the first set or the large scale? If it's a large scale, it takes at least 8 weeks from start to finish, because you have to grow the plasmid, you have to, lyse the bacteria, you have to purify the plasmids, then you have to make the RNA, then you have to purify the RNA. So I say about 8 weeks from start to finish.
And generally, follow up to that, how large is usually the team working on making the mRNA?
That's a whole, well, there are several sites involved, right? So there is a site in Andover, Massachusetts. There's a site in Michigan. Then there's a Pearl River site where we do some of the testing. So the group is several hundred people, but everybody has their own, part to play. But when you go into clinical trials, that's when it becomes, massive, because you have, all these different countries with different regulations, and we have to, monitor everything that's going on. The toughest part of the clinical trial was to get people in different time zones to interact with each other, right? Because you have people in Germany, you have people in Chile, you have people in the United States, and in the United States, you have people in Texas, some of them in New York. So, my boss, Philip Dormitzer, I don't know if he ever slept for the first couple of months, because, he was waking up at 3 o'clock in the morning to meet with this group, and then he was, another group. So it was tough to coordinate, but we pulled it off.
All right. Vidya, do we know if there's any effect of the SARS-CoV-2 on our genome? Do we have that knowledge yet?
No, we don't have any of that knowledge. I think that, you mean the SARS-CoV-2 virus or the SARS-CoV-2?
Yes.
The virus.
Virus.
I don't know about that. I mean, pathologists will have to tell you that, but it's going to be hard for that virus to have any effect on your genome, because it's an RNA virus and it doesn't have a reverse transcriptase, right? Like HIV has a reverse transcriptase. SARS-CoV-2 does not have a reverse transcriptase. So that mRNA goes straight to the cytoplasm where it gets translated, and that's it. It makes more virus and it buds out. It never gets into the nucleus. It doesn't have the signals to get into the nucleus, and it does not have a reverse transcriptase either.
Okay. And then the last one, tell us a little bit about the trials that were done by Pfizer. on its way to full approval?
Well, they were done in several different countries because everybody has got their own FDA type organization. So you have to have, first you have to have animal studies, then you have to have safety studies, right? So the first in human is a safety study. There's a small amount of people. And then you have these phase 2-3 trials that are based on what we get from those safety studies. So you can't start a phase 3 trial right away. You have to go through animal studies, then you have to look at it, then go talk to the FDA, then you have a small trial, first in human trial. I showed the FIH. That's what FIH is, first in human in May. You have to have that, and then once you get that, you go back to the FDA. You show, that if anybody has symptoms, because there were several candidates. The candidate I showed you, that was one of 4. So if people are having reactivity to a vaccine, or people had fevers, and it depends. A lot of times, some people in the placebo groups would start saying that they have symptoms, and you have to take that into consideration, right? You have to look at, okay, why did this person have, a reaction? Was it the vaccine? Was it something else? So it's not as cut and dry as it may sound, because when you're analyzing data, right, from people coming from different backgrounds, different cultural backgrounds or different racial backgrounds, it's different. Like somebody, in Guyana, right? If you cry too much, you're like, oh, you're too soft. Some cultures can take a little more pain than others. So it's subjective sometimes, and you can't, but you have to investigate. So somebody had a reaction, you have to investigate. So that's why those first in human trials were so important. So we had a way to decide what exactly was going to go forward.
Okay. Thank you, Vidya. That was the first part, the science. Now let's hear the personal. Tell us about your journey from UG.
Tell us about my journey. Oh, hold on. Let me see if I have pictures, because everybody likes to see pictures. Yeah, I have pictures. You want to see pictures, or you just want to see my journey?
Let's hear your journey.
All right. So this is where I was born. This is Wakenham Island. This is the Essequibo River here. This is Tiger Island. That's the Atlantic Ocean there. So I was born somewhere there. 77 Belt Plain. That's where I was born. That's my grandfather's house. My grandfather was a tailor. That was his tailor shop here. These are my cousins. So my nickname is actually Bobbin. So on social media, my name is Bobbin Rupchan. My name Bobbin comes from the bobbin in the sewing machine. Because my grandfather was a tailor. In the Caribbean, your nickname comes from something. So I was born there. These are my parents. This is my dad and my mom. My mom passed away last year. They were both teachers. So this is my maternal grandmother. This is my paternal grandmother. Her name is Dalari, and her name was Shahida Bakas. So that's me. You saw this picture. That's my brother. We were fishing. That's a fish right there, a catfish, a real Amazon catfish. And of course, in the Caribbean, you run around barefoot. So that's me. I was about 11 years old there. Okay, so this is how I got interested in science. Before this book, I wanted to be a journalist. I thought I was really interested in people's lives and people's stories. And then for my 11th birthday, my parents gave me this book called How and Wonder Book of Chemistry. And this book just blew my mind. And I kept reading it from page to page, from cover to cover. Almost every weekend I would read it from cover to cover. And I love the stories of these chemists who, like Frederick Waller, who challenged the vital force theory, that you had to have a vital force to make organic molecules. Louis Pasteur. A lot of these people are chemists. A lot of these, people think of as biologists or microbiologists. They were chemists. Like Louis Pasteur was a chemist, and Frederick Waller was a chemist. And so I really like those stories. And so it kind of like gave me a sense of direction. And that's my elementary school. These are my teachers from the San Souci Methodist School that I just showed you. My dad used to teach here, but he's not in these pictures. So these are all my teachers from back in 1973. That's my primary school, the newer one. Before that, it was called the San Souci Methodist School, and then it became the San Souci Primary School. So I had a Methodist education because, in the Caribbean, I went to Methodist or Anglican or Presbyterian or one of those until the government started building them. And this is my high school. I kind of grew up there. It's an Androgen multilateral school. So I think the Canadian government gave the government of Guyana a grant. So they built these multilateral schools around the country. So there were, I think, 7 of them. So I pretty much went there from 11 to 17. I took my O-levels there. This is my some of my teachers. This is my favorite teacher. So this is the first person I told I wanted to be a scientist when I was 11 years old. And she just shrugged her shoulder. Her name is Carol Argyle. And she's like, if that's what you want to be, that's what you want to be. Don't be ashamed to dream. So, you know, she's a lady who, really encouraged me in my early days because, 11 years old, you kind of think of something that you like, but, you want some guidance, which she was a very good influence on me. I mean, she was one of the best things I ever seen in my life. And that's Cheryl Argyle now. That's, she still stands the same way, all cool. She lives in New York. These are some of my other teachers, Jennifer Lord Backer, Miss Hazel, and Miss Manget. So they were teachers who lived in the dormitory with us. So they were my resident tutors, basically. So I grew up around. And these are Miss Applewhite and Miss Hardin. They were the cooks, and they were the ones responsible for taking care of us there. So I spent a lot of my years as a teenager around these ladies because I was away from home for all of the school year. So I said 9 months out of 12, I was around Miss Hardin and Miss Applewhite. And, I'm very grateful, for their guidance during those years. And that's Miss Applewhite's son, John. These are some of the other people who worked on the campus. This is Miss Lunk, great lady. This is one of the guards, Mrs. Seymour. This is Desiree. And right here is Charlene Garraway. She's a classmate of mine, so that's why she's in this picture. These are some more of my teachers, my English teacher, my agriculture teacher, my science teacher, Balwant Bagwandin. More teachers. Pamela Lord, she was a Miss Guyana. She was like 4th runner-up at the Miss World, I think, in the 70s. These are more teachers. I'm showing all these teachers because I don't want anybody to get upset that I didn't show their picture. Because the last time I did this, I got a lot of flack. Like, why isn't my picture there? So I'm showing everybody's picture, at least the pictures I could have. These are more teachers. This is my chemistry teacher here, right? Mr. Rojini Abbas. So this lady demystified organic chemistry for me. I think without her, I'm gonna have a tougher time with organic chemistry, but she really, took all the mystery out of organic chemistry for me. And that's her sister, who was my English teacher. And this is Mr. Ramjas, who was another English teacher there. These are more other of my other teachers. This is my math teacher, Jean Holder. She lives in New York. And that's me, graduated from high school with my classmate. That's me, so skinny I was. That's when I took our O-levels back in 1984. And our CXC. This is my university. This is the chemistry building. I wonder if Dr. Tinto remembers this building. I'm sure Dr. Trotz remembers this building, and Dr. Green knows this building very well. But this is the chemistry building. These are the chemistry labs up here in the bio lab. So I spent most of my years at UG. These are, there's me with some of my UG friends. All of these guys are scattered around the globe. Yeah, I'm coming to America. That's me there. Coming to conquer COVID. I didn't know what a virus was then. I knew organic chemistry then. I didn't know anything about viruses then. But yeah, I'm full of hope. That's the last time I saw Dr. Tinto, 31 years ago. That's where I got married. I know my wife from high school. So we got married in 2000. That's me not so long ago. My kids. My Rachel just turned 18. She went off to college already. Okay, so before COVID, I used to work on other vaccines. And one of the vaccines we worked on was HIV. So this was the NIH Advisory Committee. And that's me right there. There's me. There's my boss, Melinda Sidhu. There's Susan Seattle Brown. She hired me. This guy, Emilio Amini, some of you might know him. He's a big shot at the Gates Foundation. So let me show you. Let me see. Who else you should know? This here is Norm Letvin. He was a big HIV guy. This is Barton Haynes. He's a Duke. He's a big COVID guy now. So this basically was the US HIV fraternity back in 2005. And we used to meet all the time. We had a DNA vaccine candidate that went in the clinic, but it didn't work. But we did a lot of DNA vaccine and chimeras for HIV back then. So before HIV, and I'm very proud of what I did with polio. Nelson Mandela with his Kick Polio out of Africa, and we were part of it. Our site was part of that before it became Kick Wild Polio out of Africa, because the whole initiative started around 1988 because Africa was neglected in the fight against polio. I mean, I don't know how many people realize that in 96, there were 75,000 African children being paralyzed with polio. Not 86, not 76, not 66, 96. I mean, in Guyana, I mean, polio was gone by the 70s. But, you know, in Africa, I think a lot of it was due to conflict. But what Nelson Mandela did, and which is commendable, and it served as a model, he got the Rotary International to form a network of refrigerators and distributors where people were bound basically by their word not to attack these places, to leave these polio centers alone. So he got an honors thing going on with a lot of these groups that were conflicted with each other, but they all agreed that the polio vaccination program was going to work and they were going to be part of it. And that's how this disease we were able to get rid of this disease in Africa. And I think Africa, you know, the only polio you have in Africa is the vaccine associated cases, and soon that'll be gone. But you know, Africa was declared wild polio free in 2020, and that's a lot of it is due to what Nelson Mandela did. And I think it serves as a model for the future, right? For the future of Africa, the Caribbean, and Latin America. You need leaders like this to step up, you know, and form these networks and be part of supply chains and to ensure that there's distribution of vaccines. And polio is a glaring example of what can be accomplished when you get, you know, everybody to work together, including people who are fighting with each other. You know, have a polio holiday where they don't fight anymore. And these are the vaccines I've worked on. I'm coming to the end of my talk. I probably went over. So I worked at polio, which I'm very proud of. So I gotta say this: if COVID hadn't happened, and I had come to the end of my career, I would have been very proud of what I've done. You know, I found fulfillment in this job. I found purpose in this job. You know, and I've certainly handed my knowledge down to the next generation because I'm closer to the end of my career than I am in the middle. So I worked in polio. I worked in RSV, respiratory syncytial virus. I'm sure Dr. Landis knows this virus, and we're in a clinic with that. I worked in hep titer, which was a terrible disease in the 90s, and we were able to bring the rates down by 80%. I worked in rotavirus. I worked in Prevnar, which is given to kids for inner infections. I worked with HIV, and that didn't go anywhere. I worked in influenza, flu mist, and I still work in influenza, but a different vaccine candidate. I work on HCV, hepatitis C. I worked in CMV, which causes birth defects, human cytomegalovirus, and I worked in Clostridium difficile, which is in the clinic, and I worked in SARS-CoV-2. So that's basically my journey so far and how I got here. I started out, you know, liking chemistry and, you know, getting a degree in chemistry and following, you know, this path of vaccine development. And I've really found my passion. And I think, you know, that's one of the blessings I've received, that I found something I really like and I enjoy doing. And I've gotten the support of my family and my teachers, you know, over the years. And I've seen what vaccines can do in reducing diseases. And I think the most fulfilling thing I find is when I see the numbers go down, when I see the incidence of inner infections go down, or meningitis, or polio, or COVID. I get a sense of fulfillment from doing it. And it really energizes me. All right, so that's my story. I can take any questions. I can talk about how big that fish really was.
Lydia, thank you so much for your willingness to share your story. It was an enthralling story. The take-home message I heard was that the Caribbean, right here, we can adequately train our future students, our future scientists, and that training can take us onward to make an invaluable contribution on a global scale. We're going to hold on questions video until the very end, because I know that there are going to be some questions by our students that are listening about that whole transition from UG undergrad chem into the master's program and beyond. So audience, keep your questions handy. But when I was listening to that story, I did find myself thinking about the personal challenges faced by us scientists in the faculties of science and medicine across the region, and the limitations that are encountered in research labs, which don't usually lack for ideas, but are far too often plagued by aging equipment and scarce resources. So let me now turn to Dr. Paul and invite him to tell us more about the importance of science to Caribbean societies, the needed governmental support, and the H3E initiative, which is tasked with building capacity across the region. So Dr. Paul.
Thank you. Thank you very much, Madam Chair. And let me say, first of all, thanks to Professor Eddie Green and the team for the invitation to share a bit on the H3E Caribbean Initiative. And I also want to say what a pleasure it is to have shared the podium with video. Very inspiring story. You know, really, this is something that I think we can take away and you know, it's a part of a dream, but it also inspires us to do more great work like he has been doing. And I like that he emphasizes teachers so much and those who have supported him on his journey. So we're talking a little bit in a few minutes about an initiative that started not too long ago. We've called it the H3E Caribbean Initiative. And I want to give you a little bit of the background to it and to illustrate some insights as to what it is about, but also what it poses for the future for our university, our region in the Caribbean in terms of health research, and see how we can tie that back into a theme of vaccine production and the role of the university. So human, hereditary, environment, and health in the Caribbean, H3E Caribbean. This is a regional initiative. It was announced in April of this year by the Deans of the Faculty of Medical Sciences at the UWI. I had the pleasure of being involved in that then. I was then Dean, in collaboration with the Jamaica Cancer Care and Research Institute called JACRI and the Harvard MGH Center on Genomics, Vulnerable Populations and Health Disparities. So these are the co-leaders of the initiative, Professor Simon Anderson, who is at Cave Hill, myself, Dr. Alexandra Shields, who is an honorary professor at UWI, but also leader of the Harvard Center on Genomics, Vulnerable Populations and Health Disparities. And we have Leo, our operations manager, project manager. So this initiative is really, we conceived it with the vision to help us to understand the social, environmental, and biological underpinnings of disease. And pretty much when you hear videos talk, it really has that vision in mind of understanding what's happening with us in the region in terms of health and disease. And that's not a new vision for the region. The UWI's vision is tied into this. And so we are really carrying through with, if you may call it an old vision, continuing to live that vision. But a specific mission of H3E is to ensure representation of Caribbean persons in cutting-edge global genomics and health research. And we feel that mission is important because currently what we see is that our Caribbean people, the islands and the countries of the region, including Guyana, are not well represented in terms of the cutting-edge global genomics research. So when we get a treatment for cancer that is based on genetics research, that the underlying genomics population that is being studied there is not, our population is not represented. And we want to see the region represented as much as possible in terms of therapies and treatments for diseases that have a foundation in genomics and genetics. So that's the vision and mission of H3E Caribbean. I want to share with you just really what we have been doing in this project, in this initiative so far, in the few minutes we have. Now it is modeled out of H3E Africa, which has been a very successful initiative with a similar kind of mission in terms of building capacity for genomics research. And we felt that we can learn a lot from what Africa offered the continent in terms of its work in this area. And so we have modeled it, but we have added the environment, recognizing how important the environment is in the causation of disease. We have added that as a key area along with the genomics initiative. So we have human, health, hereditary, environment there as a part of the picture. And what we hope to achieve, it gives us the opportunity in the region to understand and assess our unique genetic patterns in the Caribbean region. We know that we have a very unique and interesting genetic history, let's call it that, in the Caribbean diaspora, but that ties in with the wider global diaspora. We want to understand that in terms of the role it plays in disease etiology. We want to contribute to social justice in the Caribbean by expanding the issue of equity among the discovery data sets used to develop the next generation of treatment. So this is the equity issue, the social justice issue. We want the footprint and the voice of the region there when the new drugs are being made and the new therapies are being made as we go forward. And we want to look at the unique and distinct physical, environmental, and sociocultural influences in these small islands through accurate genomic profiles of disease, population, and health. Now, I put this in here because I think this ties in well with both with what Vidya spoke about and what part of the mission that we have and the vision. This is an article that was just published just last month, hot off the press, co-authored by our very own Professor Peter Figueroa, who is a leader in epidemiologists here in the region, in the Caribbean. And he and colleagues, as you see, Peter Hotels and so on, other distinguished scientists. And the title of that paper is Achieving Global Equity for COVID-19 Vaccines. So we're talking here about equity for vaccines. Vidya gave an excellent discourse on the science, and he spoke a bit, if you listened, about the equity issues in the background. What these authors are saying is that what we need, three things. We need stronger international partnerships. We need greater advocacy and solidarity. So as we talk about the science of the vaccine, we want to put in the context, the broader context, these three critical factors of partnership, advocacy, and solidarity. And just to tie that in though, with the H3E Caribbean initiative, the direction we are going is very much in that direction. As we take on the science of the region and the building science capacity, we are very much looking at solidarity, partnership, equity issues. So we have over 100 faculty, researchers, practitioners throughout the region, currently working on 12 working groups to put a white paper together. And we're doing this in collaboration with our Harvard colleagues, I'll show you in a minute. We have partnerships across other universities. It's what I call a comprehensive partnership model. One UE, the one UE vision is there, supported by our Vice Chancellor. With the university to university academic collaborative frame is there. We have partnership with Harvard, Glasgow, UE SUNY partnership is there at the table. We're building public-private partnership. We have had consultations with the banks, the private sector, the government sector, going right through building these partnerships as we plan for this capacity building of genomics research. And civil society is at the table, Jackery itself, and working with a community in partnership to develop this model. So at the end of the day, what we're seeing is that we're aiming to have capacity building that is cross-cutting. This partnership model is driving a focus on capacity for genomics research to support novel therapies. So, and I put inclusive of vaccines. We spoke about cancer therapy, but it's not just about cancer therapy. Genomics research capacity by building things like electronic health research systems, bioinformatics systems, the laboratory support is going to put us in a good place for the next pandemic, God forbid. But even the ongoing research that we have, even before, pandemic or no pandemic, we need to look at the disease patterns, we need to look at epidemiology. And by building this capacity, we expect that we would be in a stronger place in the region to support the indigenous problems and to contribute to the global landscape in terms of research. So the point I want to make too is that it's not only the technical science that we're looking at, but I call it the macro framing of the initiative. And that's important in terms of the partnerships to ensure sustainability and equity in the long run. So if the university is going to have to step up to the plate as we're doing here, we have to do it in a comprehensive, integrated manner with multiple stakeholders. It's going to take a lot of work. It's more than putting the lab in place. We have to have those partnerships with public-private sector and have a lot of cooperative work going along the way. So it's an excellent example of academic industry partnership fostering research for development. We expect that the benefits of H3E Caribbean will bring opportunities for global research, will put us at a table as equal partners with our colleagues in the US and Europe. I know Vidya would have worked with a lot of universities through his work to get this work done at Pfizer too. We want to be there at that table to be able to partner in a very easy and open way. And we want to contribute at the end of the day to sustainable development. Because at the end of the day, what is all of this science about? What is all of the research we're going to do in the labs? It has to redound to the benefit of society so that we see the development of the Caribbean region. That is the vision and mission of H3E Caribbean, as it is for the University of the West Indies. So colleagues, I thank you for this opportunity to share. Madam Chair, I hand it over to you. Thank you very much.
Thank you very much, Dr. Paul. I very much look forward to hearing more about the progress of this initiative. Ladies and gentlemen, for our final presentation, I would now invite our Pro-Vice Chancellor and Principal to tell us more about where the UWI lies in the search for greater scientific knowledge and what our major contributions have been. And I also invite him to interrogate where we are best placed to make a significant contribution going forward. Professor?
Good. Well, thank you very much, Dean Badnock, as I know you. We're very proud at Cave Hill. Janice Badnock is our first female dean in a faculty of science and technology. So thank you so much. It was really wonderful hearing Vidya talk about the discovery of the Pfizer vaccine. And I'm glad he mentioned that this did not come overnight. And this has been, of course, a narrative that persons who are hesitant of vaccines are saying, but how is it that we were able to produce a vaccine in a year when normally it takes 3, 4, 5 years, 10 years, whatever. And of course, they were working on it ever since SARS, which was the first time that a coronavirus jumped from an animal population into a human in 2003. And I should mention that their competitor, AstraZeneca, similarly was working on their version, which was an adenoviral vaccine. And just before the COVID-19 sort of fell across the world, they actually published their vaccine against MERS, which was the second of the viruses that jumped from camels into humans in 2012. And so they had been working on their vaccine approach also for 9 years, and then they were ready when SARS came, when SARS-CoV-2, which is COVID-19, came. So again, the narrative is that those drug companies that were able to bring the vaccines in what appeared like record speed, they had been working on coronavirus vaccines for 10 years or so, or more. So in terms of our university, well, our traditional strengths have always been in medicine. We started 73 years ago in Jamaica as a medical school, and in public health and child health is where we're very well known for our research. So the Tropical Medicine Research Institute in Jamaica, the work that they produced in the 50s and 60s was really able to underpin the science, and this really was hardcore chemistry as well, of what affected refeeding programs of children who were suffering from acute malnutrition. So if you enthusiastically feed a malnourished child, very oftentimes they will die. And so they worked out exactly the sequence in which you do the refeeding. Those have underpinned the WHO's feeding programs in famines ever since the 1970s. That all came out of UWI. And the university is also very well known, of course, for climate research. So the 1.5 to stay alive was coined by Charles Taylor. And of course, we've had a number of Nobel laureates, including at Cave Hill, in the work of the IPCC, the Intergovernmental Panel on Climate Change. We've also had the tradition of research in engineering. That's traditionally been based in Trinidad. Also marine bioscience. So, you know, how could you forget that amazing discovery a few years ago by Judith Gobin and her colleagues? They went 1 mile down where on the seabed where you would be extracting oil, and they found these things called cold seeps, and around these cold seeps was an entire ecosystem of undiscovered species. And one of the 6 new species that they discovered was an octopus that, if you can believe this, it was maroon colored, just like the West Indies. So, so marine bioscience has always been a great area of research for the university, because we do, of course, have all this biodiversity in the Caribbean Sea. Environmental science is very important. So we have the center, we have CERMES, which is the Center for Environmental Research in the Cave Hill campus. And what you have heard, of course, is that we have natural products research. So this is the organic chemistry, the natural products which have come out of this region. So one of them, which came out of the Mona campus, from an extract from the periwinkle flower, is actually the cornerstone of all cancer treatments in the world, which is vincristine. So that was discovered from a plant in the Caribbean. And what this demonstrates is that over the 73 years of our university, we have evolved a full wingspan of a full faculty research university. And I'm sure you've all been reading that the university is now ranked by the Times Higher Education, which is the premier ranking agency for global universities. And we are ranked in the top 1.5% of universities in the world. But you know, what has driven that research has been, what has driven that ranking has been our research. But in my opinion, research is really only half of the equation. You have to translate that research into benefits and value for society. And this is where, you know, we heard about the kind of teamwork that's involved in bringing benefits from research to society. You heard it mentioned by Tom and Paul as the guiding force behind the H3C consortium. And when COVID struck, the University of the West Indies formed a task force. If I just share my screen quickly. Can you see my screen?
Yep.
I'm assuming you can.
Yes.
Okay, so we were immediately seized at the importance of this viral epidemic. The very first Vice Chancellor's Forum that we held was on January the 29th. I would remind you that that was well before the declaration of the pandemic, which was on March the 12th. Now, we were able to, I think, move quickly because we have a center in software engineering, which is actually part of Dean Badnock's department in Suzhou in China. And of course, as the pandemic unfolded in China, we were learning from our colleagues in China as to what was happening, and we were getting ready. So when we formed this task force, it became immediately apparent. And in that January 29th, 2020, so that's last year, my theme of my presentation was that we were facing 2 pandemics, or 2 epidemics as it then was, a virus epidemic and a misinformation epidemic. It was already in full swing within the first few weeks. And so we very deliberately set out, as you can see from the statements by the Vice Chancellor and myself as chair of the task force, that we set ourselves out to be the stall for accurate information. So when you've grown tired of listening to people like Del Bigtree, who's the king of the anti-vaxxers, and you grow tired of, oh, maybe it's 5G, or, maybe they're putting transistors in us and monitoring us, well, actually, I think they already monitor us and we do it ourselves. When you get tired of listening to that kind of anti-vax foolishness, just come to us, because you will know that you will actually get accurate information from us. And we set up a task force. And remember what I said about the wingspan of this university? This isn't just virologists. This is everyone that you need to tackle this epidemic. So I actually like this slide, which was put together by Rhonda Jaipal-Ogaro, who is one of our task force members. She's actually the communications expert. And it always makes me laugh because it looks as if the entire slide is being illuminated by the smile of Professor Maynard, who is a clinical counseling psychologist here at the Cave Hill campus. So Donna, your role and those of the other members who form a psychosocial unit, such as Professor Wendell Abel over at the Mona campus, Khatija Khan, who is on the St. Augustine campus, Miss Cecile Minot, who has a lot of experience in counseling online because she's from the Open campus where we do online teaching. And lo and behold, her counseling approaches are now being used across the entire universe, as we call it, because, you know, we were all sitting at home needing counseling. And then, of course, we have a virologist. Many of you would have heard of Professor Christine Carrington. So she's monitoring the variants as they emerge across the Caribbean. Those of you who would be from Jamaica would maybe know of Dr. Carl Bruce, who's the chief of the University Hospital, Joshua Anzinger. He's just published a paper looking at how the levels of antibodies are being affected over time after vaccination. So we're doing primary research in that area as well. Then we have our veterinary virologist, Christopher Orr. It's very important to have someone like Chris in the team, because this is still an animal virus. And, you know, he actually studies coronaviruses. All of us are now these such experts on coronaviruses, but he really is an expert. And so, you know, I remember quite early, I think in February of last year, someone said, oh, you know, there was a coronavirus discovered in a tiger in New York Zoo. Is that possible? So I called up Chris. I said, Chris, is it possible for a tiger to get? He goes, yeah, absolutely. Felines quite often get it. So, you know, having a real coronavirus expert on the task force has been essential. Then you would have a pulmonologist. You know, this is a respiratory disease. So the leading pulmonologist in the Caribbean is Professor Terence Seamungle. And then in terms of, we have Julie Meeks in terms of her health research from the Open campus. Of course, it's affecting the way that we live, the way that we educate. So we have an expert in continuing education, Dr. Veronica Simon, who's from St. Lucia. We have Michelle McLeod, who is an expert in hotel and tourism management. And she's been extremely active in trying to reimagine how we can get out of this pandemic with a strengthened tourism product. And many of you might know Don Marshall, who is, he's involved in international relations and economics. You know, this has very big socioeconomic impacts. And Anna Perkins, with whom I authored a book on the ethics of the coronavirus pandemic. She's an ethicist by profession. And so, you know, when you look at all the people that we have, we can look at the way in which this pandemic affects different parts of society differentially. So we know, of course, that it has affected females more than males in terms of the socioeconomic impact. And we have Gabby Hussain, who is a gender expert from the St. Augustine campus in Trinidad. And then finally, our newest campus, which was only 1 year old at the time, the very first guild president of the Five Islands campus, he was our youth voice on the task force. And when this broadcast is eventually made, it will be Janet Carew, who is the head of UETV, who will be putting it out. And she was on the task force. And our long-suffering registrar must be the busiest man on the entire planet, I think. He's an ex officio member of the task force. And we have Harold Watson, who's an expert in emergency medicine. And unfortunately, he is extremely busy right now trying to deal with the wave of infections and emergency care and unfortunately death, which is affecting the Caribbean. So this was the task force, and had the deliberate intention to help the Caribbean through this pandemic. So the task force coordinated UWI's internal readiness. We were well over a month ahead of the pandemic before it reached our shores, and we've helped to inform the Caribbean's response. And the way that we did this was we based it on the Zika task force, which was the first time that we had taken the kind of task force that we used to put together for things like recovery from natural disasters, like Hurricane Erica when it struck Dominica. We made a task force there to help in terms of the engineering, the safety of buildings, water quality, etc. And so this was the first time we did the same kind of task force for a medical emergency, which was Zika. And we learned from that. And then we were able to really hit the ground running 4 years later when the COVID-19 task force was put together. And it really helped, I think, CARICOM and all the various organs of CARICOM, that they knew how to interact with us. And we could speak with one voice to the CARICOM and, for example, the Council for Human and Social Development, which Eddie Green would be very familiar with, both health and education. And although we were coming with all of our expertise, they were able to interact with us through one voice. And really, these are meetings which I really treasure. And I think this has shown a level of collaboration in the Caribbean that I've never seen. I think it's really wonderful. And I would maybe single out one organization which has been so integral in coordinating this emergency response, and that's the Caribbean Disaster and Emergency Management Agency, which is based here in Barbados under the leadership of Disraeli. And so we just regard COVID as another emergency, and many of the same responses, where you have disruptions after a hurricane in terms of supply chains, in terms of communication and things like that. Many of the disasters are actually, responses are very similar. And so I think that is why the Caribbean was able to respond reasonably well. And so through the university, we provided detailed information for the Caribbean, because the Caribbean always finds itself left out. You find us in various groupings like Latin America and the Caribbean, one of my favorites, or the ACP is another grouping, Africa, Caribbean, Pacific. Sometimes we're in the Americas, like for PAHO. Then, of course, we have all of our own groupings, CARICOM and the Association of Caribbean States. And it becomes very complicated to actually find information about the pandemic on the Caribbean. So we saw it as our job to respond to the countries of CARICOM and the associate member states, who are also all of our countries that support the university. And you can see that we were able to contain the pandemic initially. And of course, now you see particularly the Delta variant causing a lot of cases around the Caribbean. And unfortunately, that's translating into deaths. And so I think I would just, I think I will wrap up here because we've been going on for some time, but I would like to make the point that when you look at it objectively and you compare the deaths per million, which is quite an accurate measure where you can compare between small countries and large countries, that the Caribbean has done very well. And we have done very well in relation to our neighbors to the north, in North America, and in relation to our neighbors to the south. And I think it is because we were able to replicate all the necessary components through really quite good coordination, which are required to build a strong health systems framework. And so I do wish to let Caribbean listeners and the Caribbean diaspora know that I think the Caribbean has handled this pandemic with great competence. Thank you.
Thank you, Professor. Ladies and gentlemen, it has been quite a night of discussion. We began with the science of COVID and the Pfizer-BioNTech vaccine, and then we migrated into conversations about capacity building and the role of the university in advancing or globally recognized science? I think we have maybe time for about 3 questions, and then I think we're going to have to wrap up. So I have one for Vidya. I'm going to actually link to NVIDIA, and this is coming from our students. So you said you were an organic chemist first and foremost, and then migrated into immunology and then into chemical engineering. So how about that transition? Was it difficult? There's not much biology in a chemistry degree. How did we go through that process?
The Guyanese curriculum actually allows you to have a minor. So I was a chemistry major, biology minor. So I had 3 years of biology. So that's why I think, in addition to that, I also, the University of Guyana had a very strong mathematics influence on the chemistry program there. So we had to do differential equations and calculus as part of our chemistry program there. So when I came to the United States, I realized that our chemistry program there allowed a very broad set of options depending on where you wanted to go in your career. So you could go into chemical engineering, you could go into biochemistry, you could even go into medical school if you wanted to do that. But I think I had a very solid foundation at UG, which allowed me to, go to Columbia and take on all of those hard classes that they claim are hard. But I had a very good applied math background at UG, and also a very good biology background from UG.
All right, I have one more for you quickly. Are there opportunities for MSc and PhDs to enter into corporate R&D? Are there still a lot of opportunities for that movement?
Yeah, so absolutely. So it depends on where you want to go long term in your career, right? Because now, what people are doing, and they're having these hybrid careers. They have chemistry degrees, but they start doing bioinformatics, or they have mathematics degrees and they start doing artificial intelligence. So there's a lot of flux now on where people want to go. I think that in this time, where we're entering this fourth industrial revolution, it's up to people to decide where they want to go and how they want to mold their careers. Because there's always classes you can take. There are always opportunities for you to learn and to grow. But yeah, there are opportunities. There are always going to be opportunities, but it's going to be up to the individual what they do with their life. And I think adaptability is very important. And I think for me, I realized that biotechnology was fresh, and I really grasped the opportunities I got, and I tried to make the best of it. But like everything else in science, you have to stay current. You have to be able to upgrade your skills. You can't just get a degree and say, okay, I'm going to cruise from now. It doesn't work like that, because science is constantly changing, especially in the corporate setting where you're competing with everybody else.
All right, I think I have one last question, and maybe I'll throw this either out to Professor Landis or Dr. Tomlin, and this is our last one. How do you see the partnerships, especially maybe Dr. Paul? How do you see the partnerships moving forward? This is a question in the chat. When it appears that so many professors are shooting for patents rather than open scientific publications. This one starts to get into policy. Anyone wants to take a stab at that quickly?
Well, I'll make a quick comment, and then I think Prof. Landis can say something. I think the patents are important because you're going to have discovery. You're going to discover stuff, and you want to protect that for a particular period of time too, until you can translate it into some economic value, which is also a goal of the university. Getting the conversion of the science and the knowledge and the discovery to impact in a real way, and also to bring benefit from a economic development point of view. And I do think having worked with colleagues across the university and other universities, I think there is room for collaboration, and yet in that collaboration and partnership to be able to protect evolving patents and emerging patents. So that will be my comment in it. I'm not sure if Clive has a view on that.
Only briefly to add that we underperform in terms of the number of our patents and translating patents into products. So I would like to see more.
Well said. I'm going to take moderator's license here, and just one last one, Vidya. This is coming at you. How does the COVID-19 vaccine affect the immune system?
Well, the vaccine by definition produces an immune response. So it stimulates your immune system. That's it. There's nothing else to it. It produces B cells and T cells. So T cells become killer cells. B cells produce antibodies. And that's the end of the vaccine. That's the story. That's how a vaccine works. So it was created to affect your immune system by it's a positive effect on your immune system, because your immune system now produces antibodies and T cells, which will protect you. So it's not innocuous, because it wants to have a response, because you don't get protected unless you have a response.
The only thing I would add is that many people say, well, you know, I can boost my immune system with vitamin C and other natural products. And that's true. You can boost your immune system in many ways. But the difference with the vaccine is you're getting protected specifically against that pathogen, which is COVID-19. And nothing does that like a vaccine.
All right. Panelists, I want to thank you.
Well, can I say something? I just want to say
Please,
one
Vidya.
last thing, because
Go ahead.
part of the discussion was about the role of the university. And I think that, and I'm going to say this because, you know, I'm biased towards natural products, but I think we seriously have to consider like an antiviral program in the Caribbean with that swath of natural products that we have. I think if we had antivirals at the start of this pandemic, it would have been a totally different trajectory. So it's something that, you know, the University of Guyana, University of West Indies, they need to start thinking about and reaching out to possibly, you know, some other institutions, or probably better institution to institution, because companies are not going to get involved unless they see something. But an antiviral program in the Caribbean using natural products in the Caribbean, like these high throughput screening. I saw there were some virologists in the Caribbean. I mean, certainly they would know who to talk to. But I would think that's a place that we could start almost immediately, because we have that history in natural products. All right.
Thank you, Vidya. That is food for thought. Lots for us to take on board. And I want to go ahead and thank the audience for their questions. I apologize that we weren't able to actually answer all. And I want to thank you, panelists, again for your time. I am going to turn this back over to Professor Green for the wrap-up. Thank you, everyone.
Well, good evening. I thought that my colleague, Dr. Rennie, would have wrapped up. So I think we will join. I will just say, I really was intrigued by the discussion. I heard video before when he made his presentation to the University of Guyana. Tonight, however, he excelled in explaining the science, and the science so much linked with the future of Caribbean universities. What we heard from the history, we heard from Professor Landis, the partnerships, and the future of the H3 EC is so intriguing to me. But I believe most of all, I hope the students who are there have really learned a lesson of perseverance, a lesson of commitment, and a lesson of looking to the future in aspiration and with sense and sensibilities. I thank you so much, Chair, and I want to give the last word to Dr. Rennie, if you don't mind.
Thank you for that. I don't think there's much more for me to say. Thank you so much, Professor Green, for doing an excellent wrap-up. It just leaves me to say how proud I am of the tradition of the university. I heard for the first time, I'm not in the natural sciences or science and technology or immunology or virology. I heard some of the things that brought me back to my organic chemistry days that I haven't heard for, I won't say how many years, but I must say that it was a great opportunity to showcase the Caribbean through its universities, the work of Professor Landis, the work also of Mr. Tomlin Paul, and our guest star tonight, the work of Vidya Rupchand, who through his story, his journey from the Caribbean to Pfizer, has shown us and our students really what is possible, and really giving so much accolade and so much appreciation to his teachers. My mom's a teacher, and I know teachers often go unrewarded for all of the inspiring messaging, not only from the work that they do, but also to encourage their students to reach to the heights. I would like to thank Professor Green from the University of Guyana for reaching out to us here at the University of the West Indies to host video. And I would like to thank my moderator, our moderator for tonight, with her impassioned moderating, brought a lot of life and energy to this panel. And to our panelists, Professor Landis, Dr. Tomlin Paul, and of course, Vidya and Pfizer for allowing Vidya to speak so eloquently to us about his journey. And all of those of you in the audience, I know that you have so many choices for webinars, and just that you have stayed the course. I always look at how many participants start and how many stay the entire course, and you have stayed the course. We will try to make this video available on UETV and also on our website at the SRC. So those of you, and I will ask also Dr. Dean Badnock to make it available on her website, as well as at the University of Guyana, if that's possible, since we're all co-collaborators on this webinar. So thank you also to our IT team at the University of the West Indies for making this possible. And without further ado, let me bring tonight's events to an end, and thank you all for joining us, and to our wonderful panelists for bringing so much information and inspiring stories to this audience. So thank you, everyone, and have a wonderful evening.
All right.
Thank you, Chair.
Thank you.
Thank you, everyone.
Thank you, Vidya.
All right, no problem. Anytime.
Bye.
Thank you, Vidya. Thank you,
All
everyone.
right, thank you.
Good night.
All right.